Thank you so much for the introduction, and everyone for logging in to listen. So I'm sure we can all appreciate in this section how incredible sexual selection is and how it's played a role in the evolution of some of our favorite, most outlandish traits and behaviors across many different taxa. And so knowing this incredibly powerful evolutionary force that it is, often the next question that we have is what actually limits it? And we have many examples of how natural selection can directly constrain the size or degree of a sexually selected trait. But I'm also interested in how more general environment or abiotic conditions can exert a natural selective force to limit the scope of sexual selection by acting not on traits that are not themselves the direct products of sexual selection. And so ways in which the abiotic environment can shape organisms include, of course, temperature, and there are many well-studied examples of how the clear physiological consequences of extreme hot or cold temperatures influence organisms. But another abiotic factor that can shape organisms is precipitation, and we're accustomed to thinking about a lack of precipitation, whether it's a desert or drought conditions, can shape organismal fitness. But too much rain can also influence organisms. And this might be particularly relevant in tropical environments where in a typical tropical rainforest temperature is quite constant throughout the year, and instead it's precipitation, which varies dramatically from months in which there's a lot of rain falling to periods of the year that are much drier. And we know that this has a lot of influences on the plant communities in these places, but increasingly we're starting to realize that this also has consequences for the endotherms in these habitats, whether it's through direct effects on survival or indirect effects of thermoregulation, food availability, and fecundity. So this is the focus of my work, is understanding how rain affects sexual selection using our favorite poster children, the mannequins, which inhabit a range of tropical precipitation regimes, and of course have their lek mating system in which males are gaining all of their reproductive success from the displays that they do at designated display sites. And there's a lot of inherent reproductive skew frequently and social hierarchies at these display sites. And in many species, including the one that I'm going to be talking about for the rest of this presentation, just getting to a point where they can have an opportunity to reproduce is in itself somewhat of a journey. So white ruff mannequins, Coropipo altera, take 4 years to reach their adult definitive plumage. And at that point, they may be subordinate at a display site, and it may still be some years before they rise in the social hierarchy to be dominant at their own display site when they can display for females and potentially mate. But of course, in some cases, females may not choose to mate with them, so reproductive success is still not guaranteed in that case. We know that rain influences the species physiologically and behaviorally. After big storm events, we see evidence of fasting and birds using stored fat to survive. We see mortality after big storm events. In one population, at least, males frequently migrate from low elevations to escape heavy rains that fall during the non-breeding season. But when these males return for the next breeding season, they often bear the consequences of this choice in that they lose their social status, and females actually prefer males that have not migrated. This population gets absolutely demonized in terms of rain per year. And we see really astonishingly low estimates of apparent survival for males in this population, considering how long it takes for them to get to reproductive maturity. And this is when we compare to another population of white ruff mannequins, where they're subject to about half as much rain per year. Estimates of apparent annual survival there are 0.9, quite a bit higher. So I'm interested in how these differences in mortality that are happening at these population levels might influence the reproductive skew and strength of sexual selection that these populations are experiencing. So we might predict that in a population with high survival, the males that rise in the social hierarchy and are attractive to females have the opportunity to survive for many breeding seasons. So in that case, perhaps that population has quite high reproductive skew as compared to a population which has much lower apparent survival, and there's a lot more turnover in the hierarchy amongst males. So even if one male is reproductively successful in a given breeding season, the following year he may have migrated, he may have died, and another male might have an opportunity to reproduce in the next breeding season. So I'm testing the relative genetic diversity of Z or sex chromosomes to autosomes. So there are basically 3 Z or sex chromosomes for every 4 autosomes in a population. And if there's no reproductive skew going on, then the genetic diversity of the Z chromosome relative to the autosome should be about 0.75. But if there is reproductive skew, that is, there are some males that are having an opportunity to reproduce, then that ratio of Z to A genetic diversity should decline to be less than 0.75. So we basically just think about male mannequins as sets of 2 Z chromosomes. In our population with high survival and high reproductive skew, we would predict that there's going to be lower genetic diversity of the Z chromosome relative to autosomes as compared to a population that's experiencing a lot more turnover in displaying and reproductive males. There's basically a higher effective population size of Z chromosomes at that site, and we predict higher genetic diversity of the Z chromosome relative to autosomes. So my question is what the relationship is between this ratio of Z to A genetic diversity and environmental variables that could influence demographics of these different sites. So to collect this data, I went and sampled in Costa Rica on a replicated biogeographic gradient. So Costa Rica is sort of divided into a Caribbean slope and a Pacific slope by the central mountain range, which I've showed with a dashed white line. And on both sides of that central mountain range, conditions go from a bit drier in the northwest to somewhat more wet in the southeast. And I've shown all of the Caribbean slope sites in shades of blue and the Pacific slope sites in shades of red. So this was an adventure in finding ways to get to the particular elevation and primary forest that these guys like to hang out in, and that's somewhat reflected in the sample sizes that we have, but ultimately we were able to come away with 158 blood samples for individuals distributed across 7 sites. And so we did a restriction site associated DNA sequencing approach to look at nucleotide diversity of Z chromosomes to autosomes. The environmental variables that I looked at in relation to that metric included temperature because of the potential for thermoregulatory costs to influence survival. Of course, we wanted to look at mean annual precipitation, knowing that there are striking differences in our estimates of apparent survival for sites that experience different amounts of rain every year. And we were also interested in how the distribution of rainfall throughout the year might affect display cost for these sites. So white rough mannequins display during the dry season, but you'll notice on the Caribbean slope, there's much less seasonality in precipitation. So there's actually a fair bit of rain that falls during the so-called dry season. And so we thought that might be have an effect on the cost of display for males that are displaying under these conditions. Our metric of Z relative to A did actually vary by sampling site, so that was an exciting first step. And just as a quick reminder, these lower values of Z nucleotide diversity to autosomal nucleotide diversity are reflective of increased skew, while higher values of Z nucleotide diversity relative to autosomal diversity are indicative of reduced skew, potentially weaker sexual selection. In the relationship between temperature and Z to A nucleotide diversity, we didn't really see a pattern. Surprisingly, we also didn't see any clear relationship between total mean annual precipitation and Z to A nucleotide diversity. But when we looked at seasonality and precipitation, we did find that Z nucleotide diversity was highest at sites where rain was less seasonal. So in these places where rain is falling throughout a larger portion of the year, we have, it looks like, less skew going on as compared to sites where rain is falling more seasonally. You may have noticed, however, that there is somewhat of a slope effect going on. So overall, all of our Caribbean slope sites are both at lower seasonality and have reduced skew. And there's a possibility that there may be some underlying genetic differentiation going on that could be affecting these relationships. And we know that Caribbean and Pacific slope white rough mannequins have previously been described as subspecies, but that hasn't been looked at genetically yet. So as a first step, I went and did an admixture analysis looking at ancestry of these individuals and did see that there's some pretty strong differentiation between these groups with no recent or ongoing gene flow happening between the Caribbean slope and the Pacific slope. So I definitely want to continue to dig more into this genetic data and try to understand some of the nuances of the biogeographic history that might be going on. But I think it's exciting that we do see this relationship, that it is suggesting, that perhaps having to deal with multiple energetic costs, both the cost of display during the breeding season and the physiological stressors of contending with rain, can influence the scope of sexual selection for these birds. And I think it's also a plug for using some of these cool genetic techniques to test predictions that we develop through behavioral observations or physiological tests and things like that. So I'm very excited to continue to play around with this data set. But for now, I just want to thank all of the funding sources, and absolutely the Mannequin Genomics RCN have been instrumental to this project. And I really appreciate everyone's generosity with their time and expertise. And of course, I have to thank the organizations in Costa Rica who permitted the collection of this data. And if I have time, I would be happy to take any questions.